Hello, everyone, and welcome to the fourth week of the Tyson Summer Seminar Series. I'm Mahal Bugay, the current graduate student in the EEPB program at WashU. Before I introduce our wonderful speaker, I'd like to lay down the housekeeping rules for the seminar. First, please make sure you're logged into YouTube so that you can use the comment feature in the chat to say hello and tell us where you're from. Second, you're welcome to submit your questions during the talk, but please be aware that the submitted questions will not be asked until the Q&A at the end. That is all the rules, so now let me introduce our invited speaker for today, Dr. Anny Chung from the University of Georgia. Dr. Anny Chung received her BA in Biology and International and Area Studies from Washington University in St. Louis in 2011. During her time at WashU, Dr. Chung conducted research at Tyson Research Center under the mentorship of Dr. Tiffany Knight. After her undergraduate studies, she received her PhD in Biology from the University of New Mexico, where she worked with Dr. Jennifer Rogers in understanding how plant-soil microbial feedbacks drive long-term coexistence dynamics between two desert grass species. Her research journey then led to a postdoctoral fellowship with Dr. Peter Adler at Utah State University. This past year, Dr. Chung established her lab at the University of Georgia in the Department of Plant Biology and Plant Pathology as the Haynes Distinguished Assistant Professor in below-ground field botany. Research in her lab focuses on plant-microbial interactions and how these interactions impact plant communities, especially in relation to community structure, species turnover and stability, and species competition and coexistence. With that being said, please welcome her as she presents her talk. And Dr. Chung, you have the virtual floor.
Thank you so much, Mahal, for the great introduction. And I am just figuratively be back at Tyson today, where, this is where I learned how to be an ecologist. And to prove, I actually found a picture of me in my vintage Tyson t-shirt from 10 years ago, catching bees for my then undergraduate thesis. So it's really exciting to be back and to share my science. So in most parts of the world, if we were to go out for a hike and a walk, the most dominant living feature of that landscape would be the vegetation or the plant community. And as we walk around, some of the things we might notice are, what are the species that are present? Why are some communities very diverse and others not so speciose? And what might allow those species to coexist with each other? And then once the species get there, why are some of these species more common, whereas others are much more rare? And the classic approach to answering these questions has mostly focused on defining key abiotic niche dimensions that are then altered by interactions such as competition and consumption. And these are questions that are fundamental to population community ecology and have been asked for decades, past century, if not longer, but we continue to ask them because we don't have all the answers. And for plant communities, I argue that one crucial piece of the puzzle that has yet to be integrated fully is interactions with microbes. And so this piece of the picture has been brought to the forefront by the microbial revolution, some might say. And with these advancements in molecular biology and sequencing technology in particular in the past few decades, we're really starting to understand the unseen diversity and majority that really rules the visible world. And so, for example, research in the human microbiome has completely revolutionized the way we see ourselves as organisms and also how we think about healthy or diseased phenotypes. And as a plant ecologist, I think it's crucial that we incorporate these advancements into ecological theory. And so today I want to specifically focus on the topic of coexistence and the role of plant-microbe interactions in plant coexistence. And so first I want to get us all on the same page. Modern coexistence theory, in the sense of Peter Tyson's work and Zuck's work in the early 2000s, organizes coexistence mechanisms in non-fluctuating environments into equalizing and stabilizing mechanisms. And to help us visualize this, here's a really kind of simple and classic model of two plants. So we have P1 and P2, which consume resources R1 and R2. So on average, there may be inherent differences in their competitive abilities. So for example, here, P2 is the stronger competitor, resulting in its positive per capita growth rate and outcompeting P1. And so one way for these two species to coexist is for the fitness difference between the species. Ooh, I can't really see my pointer. Oh, here we go. For the fitness difference between the species to be smaller, and mechanisms that facilitate that change are called equalizing mechanisms of coexistence. These can also coexist through niche partitioning. In this, specialize on different resources, R1 and R2, and this scenario leads to a pattern called negative frequency dependence for each species, where their population's growth rates are the positive and the highest when they are rare, and decrease as they increase in frequency in the community. These negative frequency dependent mechanisms are called stabilizing mechanisms of coexistence, as they are thought to facilitate stable coexistence at equilibrium. So, however, one thing that is hard to reconcile in the field of coexistence theory is that most of our models are phenomenological. And so here I use resource partitioning as an example, but there could be multitudes of potential niche mechanisms that underlie this phenomenon of negative frequency dependence that we measure to determine coexistence in a community. And so maybe unsurprisingly, going from phenomenon to mechanism requires experiments. And so today I want to talk about some of my past attempts to pinpoint plant microbe interactions as a coexistence mechanism. So how might microbes be important for coexistence in plants? So let's put this really simple model into context. Incorporating microbes, we can see that they actually moderate every single circle and arrow in the very simple model. So for example, you have pathogens and symbionts that directly affect the performance of plant health. There are resource mutualists such as mycorrhizae that, ooh, it says my internet connection is unstable. Hopefully you can all hear me still. There are resource mutualists that alter the way plants interact with resources. And then there are also microbial decomposers that alter the rate of supply of different resources available to the plants. And not only can different microbes affect all these different parts of this model, specific taxa can actually potentially change among all these different roles within the course of their life history. And so I think this really brings home the importance potentially of microbes in mediating competition and coexistence among plants. And so one way to bridge plant microbe interactions and plant coexistence is to think about plant soil feedbacks. And I know many of you here probably have learned about or heard about plant soil feedback. But to go through it rather quickly, plant soil feedbacks can be thought of as an interaction or phenomenon that occurs in several steps. So first, plants are known to accumulate specific soil microbial communities that are unique to the different host plants. And these soil microbes alter the performance of their plant hosts, often in a negative way. And plants are often found to do better microbial communities that are associated with another co-occurring competitor than their own. And this leads to a negative feedback, which we call negative soil feedback. And researchers have hypothesized that these negative feedbacks can be one of these mechanisms of coexistence. And when I started doing my graduate research, this was an idea that I was really interested in, but thought that there wasn't enough comprehensive experimental evidence that really demonstrated this idea. And so how can we actually demonstrate it? What I've done here is added microbes to our simple model. And so in nature, we might expect, so for example, resource partition to drive this negative frequency dependent pattern that we've just talked about. If microbes play an important role in generating this negative frequency dependence, then it take away the effects of microbes, then we should see that this slope, negative frequency dependent slope, become less steep. And so that is one way to quantify the contribution of microbes to stabilizing mechanisms of coexistence. And so during my PhD, this is what I thought to test out. And I spent the majority of that work in New Mexico, in the kind of southwestern United States, which covers the northern edge of the Chihuahuan Desert grasslands. And this is home to the Sevieta long-term ecological research site. And here, this grassland is dominated by two species, blue grama grass and black grama grass. And very conveniently, I will from now on use the color blue when I talk about blue grama and this darker gray color when I talk about black grama grass. And so we have really good long-term data and evidence that shows that these two species co-occur long-term. However, at the time, there wasn't a lot of research into the potential niche partitioning pathways to coexistence among these two species, especially since they're congener grasses. We might expect that they play similar ecological roles. However, just like all the other plants, they are known to interact with soil microbial communities. And within this kind of simplified system that is dominated by just two species, if we're able to target just these two species and their feedbacks, one benefit is that that can explain the majority of the dynamics that are happening in this system. And so I first used a greenhouse experiment to test the role of plant soil feedbacks in competition and coexistence in these two species. And so to test the competition part of it, I set up what's called a response surface experiment, where in each of these pots, I varied the relative frequencies and total densities of blue and black grama plants. And then on top of this competition design, I layered microbial treatments where half of the pots were inoculated with soil microbes that were collected under blue grama plants, and half of them inoculated with soil microbes collected under black grama plants. These inocula were collected that existed in a 7-year-old field model at the time. And of the soil inocula, I then sterilized half. That way we can isolate the effect of the live microbes. And so to first show you the effect of the inoculation, what we did towards the end of the experiment is to harvest all of the roots of the plants in the pot, sequence them to figure out the root-associated fungal communities. And here in this figure, what I'm showing you is each point represents the composition of the root-associated fungi found in one sample, where plants inoculated with black grama soil inocula are colored black, and plants inoculated with blue grama soil inocula are colored. The closer away the points are to each other, the more similar their root fungal composition is. And so what you can see here is that there's a very strong difference in root fungal composition between plants that were inoculated with black grama inoculum versus blue grama inoculum. So we were able to show that indeed these two species associated with distinct microbial communities, which is one of the first assumptions of plant soil feedback, but not always directly tested. And so what are the effects on composition and coexistence? So before I show you the actual results, I just want to run through some potential expectations. And so if negative plant soil feedbacks contribute to stabilizing coexistence for blue grama, what we might expect to see is first negative frequency dependence for blue grama in this figure shown by a decreasing per capita biomass going from 0% frequency to 100% frequency. So that's our negative frequency dependence. And we would expect to see a steeper negative slope under inoculation from self microbes versus microbes from the other plant species. And for black grama, we would see, again, expect that if negative plant soil feedbacks play a strong role in coexistence here, then we might expect to see this stronger and steeper negative slope going from 0 to 100% for black grama under inoculation of self microbes versus the others microbes. Okay, so that's the expectation. So what did we actually find? So excitingly, we found that for blue grama, it indeed showed negative frequency dependence, and that this negative frequency dependence was much steeper and stronger when it was inoculated with its own microbes, which supported the original hypothesis that negative plant soil feedbacks could contribute to coexistence between these two species. However, the interesting thing is that we did find that instead of negative frequency dependence, we found positive frequency dependence for black grama, with also a steeper slope under self microbes. And so I think this experiment, what I can conclude from the evidence is that stronger negative frequency dependence in the superior competitor, blue grama, could facilitate coexistence, especially in this scenario where otherwise it would just outcompete black grama, which was the weaker competitor in this experiment. And so after I did that greenhouse experiment, one thing that I really wanted to figure out, and a question that I wanted to answer was, are these dynamics that I'm seeing in the greenhouse important at all in the real world, out in the field where a lot of other mechanisms are also happening? And so luckily, because the Savietta is a long-term ecological research site, we have this wealth of long-term data that we can use. And from this long-term vegetation monitoring, there's a where for a 400-meter long vegetation transect, so you can actually see the PVC marker here for one end of this 400-meter long transect. It has been sampled continuously twice a year at 1 centimeter resolution since 1989. And from this long-term data, there's some really interesting dynamics that we can start to see between blue and black grama. So what I've done here is plotted a small section of that 400-meter transect from meter 295 to 295, then composition of relatively percentage of blue grama to black grama every 4 meters, and 1989 all the way up to 2012. So each point shows the relative composition of blue versus black grama in that point in space and time. And as I looked at this long-term data, a pattern that jumped out was there are certain patches in space along this transect where the dynamics seem like there's a lot of turnover in relative abundance between blue and black grama. So for example here, which I call kind of the dynamic patches, it kind of goes back and forth between being dominated by black grama grass versus blue grama grass. But then there are also a lot of patches where the composition stays relatively static. So for example, here is a patch that has mostly been dominated by blue grama grass throughout the entire that we have. And so I was really curious what might be underlying patterns. And one thing that I did find out after doing a lot of sampling of soil nutrients and moisture and temperature and texture is that it doesn't seem like any of these patterns are driven by abiotic differences. And so that got me thinking, could plant soil feedbacks be a mechanism that underlies the differences between dynamic and static patches, and could negative plant soil feedbacks be promoting stable coexistence in these static patches? So in order to test this hypothesis, I set up a field experiment directly next to the transect where the dynamics were happening. So I went back to the long-term data, analyzed it, and picked out patches, 10 patches that displayed dynamics and 10 patches that displayed static dynamics, and spatially paired them to take care of some of these spatial gradients in adaptive characteristics that exist along this transect. Within each experimental plot, in order to measure plant soil feedback, what I used were seedling transplants of blue and black gramas and adult resident plants that were naturally occurring to act as soil microbial donors. So what I mean by this is, so for example, here is a small blue grama plant that was transplanted next to a resident blue grama plant, which meant that it was experiencing blue grama soil microbes. And here's a black grama seedling that is experiencing blue grama microbes, and blue and black grama seedlings are experiencing black grama microbes from a neighboring resident plant. So these feedback plants were planted out encased in a kind of mesh cylinder that excluded direct root penetration, but allowed colonization from the microbes. We also planted out these kind of exclusion control plants that were planted in these cylinders that you see here that were made out of very, very fine mesh that would exclude direct hyphal penetration from fungi trying to colonize the soils, and also planted out in field soils. And so by comparing the growth of these feedback plant seedlings, we can calculate plant soil feedback. And by comparing growth between the feedback plants and the control exclusion plants, out whether the effects of microbes are predominantly positive or negative. And so, what did we find? Here, I'm showing you the results as a plant soil feedback metric. And so this metric here for blue grama and black grama plant soil feedback is calculated as the log ratio of the biomass of a plant in its own soil over the biomass of the plant in other soil. And so if this metric is significantly negative, that means that it's doing worse in its own soil versus another species soil, and therefore there's negative feedback. And so what we found was that feedback differed between the dynamic and static patches. And specifically, especially for blue grama, in static patches, it did so much better in next to black grama neighbors and in black grama microbes versus in its own microbes that we found significant negative feedback, but only in static patches and not in dynamic patches. This trend was also present for black grama, although these measures of plant soil feedback are not significantly different from zero. And so we were also, after these experiments, able to try to pinpoint what potential microbial taxa that might be underlying these particular dynamics, which is really fun because I think a lot of times it's easy to just soil as a black box. And so from the greenhouse experiment plants, which are kind of in this star symbol here, and the field transplants in the circle symbols, sequencing their roots, we were able to figure out from over 1,000 taxa a few candidates that potentially correlated with plant soil feedback in their abundance. And so, for example, we found that this Fusarium rhodolens strain was correlated with more negative plant soil feedback, whereas these other 3 taxa are correlated with positive feedback. And it actually makes sense considering what little is known about the natural history of these species. So for example, Fusarium rhodolens is a known asparagus pathogen. And so perhaps it makes sense that it is correlated with negative feedbacks, whereas species in Glomeromycetes are a part of the arbuscular mycorrhizal fungi, which are known resource mutualists with plant hosts. And by beginning to do this kind of work to try to pinpoint specific fungal or microbial taxa that may underlie plant soil feedbacks and the dynamics we see by just looking at the plants, I think gives really exciting opportunities in the future to do inoculant experiments and also to understand more specifically and more mechanistically how plant soil feedbacks work. And so from this set of experiments, what I think I've shown that plant soil feedbacks are a potential mechanism that can generate the right phenomena that we consider to be contributing to stabilizing coexistence. So for example, I found that negative plant soil feedbacks in this dominant competitor plant increased its self-limitation to help promote coexistence. And in the field, we found that more negative plant soil feedbacks corresponded to these patches of high stability, and that these patches the feedbacks can also be correlated with specific root fungal taxa. And so as I was wrapping up this part of my dissertation and thinking about what I wanted to do for my postdoctoral work, one thing that I was really interested in pursuing was figuring out the relative importance of plant soil feedback as a mechanism of coexistence versus all of the other things that are happening in the field. And so I found what I thought then was the perfect system in which to answer this question. And so this is a picture of the sagebrush steppe in Idaho, where I did this research, and it is dominated by 4 different plant species. We have free-tip sagebrush and 3 different perennial grasses. And so my postdoctoral advisor, Peter Adler, has done a lot of multi-species modeling based on long-term observational data collected about the demography of these 4 species. And what he's found is that these 4 species coexist, and they coexist stably due to large niche differences. And more specifically, when they looked at these effects, what they found was that these effects for stable coexistence, whatever the mechanisms were, these mechanisms were most important acting in the early establishment stage and at local spatial scale. And so these are figures from a modeling experiment that they conducted, where they removed niche mechanisms from different life stages of these population models. And it is when you remove the niche differences from R, which is the recruitment stage, where you see this drastic change in both the invasion growth rate and predicted mean cover of these 4 species compared to the all niches control, where coexistence then breaks down and you with a monodominance of ARTR, which is the sagebrush species. And so from all of these different lines of observational and modeling evidence, it seems like there's really strong potential for feedbacks to be that important mechanism that acts at the seedlings and at local spatial scales to promote coexistence in the sagebrush steppe ecosystem. And so, since we knew all of this about this, what I wanted to do for my postdoc was do the experiment to actually quantify the relative contribution of plant soil feedbacks to coexistence. And I wanted to do this via a field experiment. So the design of the field experiment is it's rather similar to the previous field experiment that I just showed you from my dissertation work. And here we have 3 different treatments to determine coexistence mechanism. First, seedlings are directly transplanted into the soil with no cylinders, nothing, just directly into a hole in the ground. And so this is our all niches control. So essentially that seedling would be experiencing every single possible niche mechanism that is occurring at that site. Then we had seedlings that were transplanted in a cylinder with live soils, but that excluded direct root competition. And then the final treatment was a seedling transplanted in a cylinder with sterilized soil, so that excluded both roots and microbes. We tested these treatments for all combinations of the four focal plant species, and also 5 different soil environments. So the 4 species and their microbial environments, as well as bare ground with just essentially everything else. So these bare ground tests it didn't necessarily stay bare throughout the entire growth season, but it did any of the other 4 species occurring next to it. And so again, these soil environments were tested by transplanting seedlings around a focal plant individual that acted as the microbial or soil environment donor. We recorded germination, survival, growth as our response variables, and this experiment was repeated twice, so over 2 growing seasons. And so the experiment was finished at the end of last summer. So I've been slowly working through the data, and I'm excited to share some preliminary results from this experiment. So first, I think what I can say for this particular site is that microbes are important. And so this figure here shows above ground biomass of all of the plants combined and also across both years, only separated out by the different treatments and also the different soil and microbe environment types. And you can see here the most prominent pattern that jumps out is that all plants are smallest when they're experiencing all niche mechanisms. When you exclude root competition, they are a little bit bigger, and they are the biggest when you exclude both roots and microbes. This is a pattern that's so, I guess, consistent across years and across the different plants. I mean, you can actually just see it by looking at the plants in the field. And so here we have 3 little baby sagebrush seedlings. This is the all niche control. And here's the root exclusion plant, a little bit bigger. And here's the microbe and root exclusion plant, and it's nice and big and happy. And so I think here there is a very strong pattern that these niche mechanisms or microbes and root competition are really important for the growth of seedlings and seedling establishment. However, these microbes are important, but maybe not necessarily for coexistence relative to other things. And the reason I say that is because remember from the previous work, in order to demonstrate that these mechanisms are important for coexistence, they have to act differently across the different host species. So in order to figure that out, we need to look at each individual plant species one by one. And so here's just sagebrush, for example, and its response in the 2018 season. Again, it's above ground biomass, and you can see the very strong pattern of it being the smallest under all niches, a little bit bigger under root exclusion, and doing the best when you exclude both roots and microbes. However, in order to show that these potential mechanisms are important for coexistence, we have to compare its performance in its own neighbor environment, so its own soil microbial environment, versus when it is in all of these other environments. And when I did that, what I found were only very subtle differences. And so, for example, here, the only even marginally significant differences are between its performance in its own soil and in heterostaper soil, and also in pseudoregnaria soil, where it did better there in both cases. And what's even more interesting is when we looked at the all niches control, we didn't see any evidence that intraspecific effects were stronger than interspecific effects. What I mean by that is that it's not doing the worst in its own soil compared to all the other soils, which is what we expected to find given the really strong results from the previous long-term observational data, which our all niches control should directly reflect. And so that was really perplexing. I'll talk a little bit more about why we think that might be a little bit later. And then finally, in a bootstrap extrapolation where I tried to combine the effects across germination, survival, and plant growth using this estimated biomass contribution metric, you can see that perhaps the role of root competition and microbes are not specifically to contribute to stabilizing coexistence, but here you can see that as you exclude these different niche mechanisms, there begins to be stronger differences in estimated biomass among the different transplant species, and perhaps microbes a stronger contribution to these fitness and dominance differences rather than the stabilizing coexistence mechanism we were thinking about before. And so I think that particular experiment and experience really taught me that going from phenomenon to mechanism is really difficult. You know, despite this really strong potential for seeing, or I thought was strong potential for seeing plant soil feedbacks as a really strong contributor to coexistence and potential for then trying to incorporate that into future population modeling, we found that microbes were strongly pathogenic in general, but may not be the stabilizing coexistence mechanism that we were looking for. In addition, these field experiments did not demonstrate these strong intraspecific effects that we've seen in observational data. And Peter and I have kind of gone back and forth about this and discussed it quite a bit. And one potential explanation that we've landed on is this idea of the ghost of competition present, which was kind of brought up in a paper by Chantak et al. In 2018, where it's just discussing this phenomenon we see where you generally find stronger intraspecific competition relative to interspecific competition in observational studies versus experimental studies. And his reasoning was that it might be the case in observational studies that these species that you are looking at and measuring have already sorted into their realized niches, into places where they compete the best. And therefore, the data are essentially biased towards you seeing or measuring, as a consequence, stronger intraspecific effects relative to interspecific effects. So that's something that we're still kind of chewing on and trying to figure out. And if you have any comments or suggestions or advice, I would love to hear it. And to wrap this talk up, so why should we consider plant microbe interactions and feedbacks in plant ecology? I think that I hope that the work that I've shown here today demonstrate that plant soil microbial feedbacks can play important roles in determining coexistence and stability in plant communities, but that terms and conditions apply. And for me as an ecologist, these terms and conditions and figuring out when plant soil microbial feedbacks or plant microbe interactions are important is really the more exciting part of my work going forward. And that in general, incorporating microbial perspectives could lead to new insights for global patterns in ecology and also understanding these future environmental challenges that we face. And kind of coming back to this really simple model, I hope that I've demonstrated that by putting even just a very simple model into a microbial framework, we can learn a whole lot of new things about population and community dynamics. And so with that, thanks again for inviting me, and extend a huge thank you to all of my advisors and collaborators, students and technicians, and my funding resources that helped make this work possible. And I'm happy to take any questions.
Cool. Thank you, Annie, for such a wonderful talk. And I'm sure everyone's giving you a great round of applause right now. So again, to reiterate, please ask your questions in the live chat and we will ask them for you. So one of the first questions asked was, do you know of any studied examples of mutualist microbes favoring competitors?
Yes. So I'm assuming that the question is asking about whether there are mutualist driven dynamics that then contribute to competition and coexistence. And I can think of two examples specifically. One is a paper by Jim Beaver, where they showed that these feedbacks, negative feedbacks that contribute to coexistence, are possible with interactions between plants and their arbuscular mycorrhizal mutualists, where in this case and in both of these cases, it turns out that plants associate with mutualists that end up being not as good for them compared to mutualists that are associated with other co-occurring plant species. And so the example of this that I can think of is in a rhizobium system where there are these different strains of rhizobia. This is work, I believe, by Andrew Seifert, where I think 4 different clover species and their associated rhizobia also demonstrated these negative feedbacks.
Cool. Another question was, how do differences in the fungal communities between the blue and black grama's compare to those of other plants in the communities? Are they more similar than expected or less? And then they're referring to your NMDS plot that you had earlier.
Mm-hmm. Yeah, so in that NMDS plot, that's purely from the greenhouse experiment. And so we weren't able to directly compare that to any other plant species because there were only 2 species in this greenhouse experiment. But I mean, I think there is this kind of underlying assumption that more closely related hosts should potentially have more similar associations with below ground microbes. It has panned out in some cases that I've seen. It has not panned out in other cases that I've seen. I think in this system in particular, there have been, not myself, but other folks have examined the root-associated fungi of a few other species that occur in lower abundance, and there's another grass, Boraphis jamesii, and there's also creosote bush that also occurs. And in those cases, I believe that the results show that blue and black grama do have slightly more similar fungal community compositions compared to these other species that are also there.
And then I think related to that, if you tossed another fungal community in the plot, how far away from the ones you show might it be?
Oh, well, I mean, it really depends on what fungal community you're tossing into the pot, right? Logically, it makes sense to be testing taxa that are there in the field, right? We're not, synthesizing a make-believe community, right? But there could be very strong differences. And kind of to interpret this question slightly more broadly, there are really cool studies that have shown that different soil inocula can completely change the trajectory of a prairie or an old field restoration and succession, even if you just introduce it just once in the beginning of the restoration process.
Another question was, between the two grama grasses, does stronger negative plant soil feedbacks in blue grama cause blue grama to be more rare than black grama at a local or regional scale?
That's a really good question. And so the answer is it's complicated. So I didn't include in the talk due to time, but so when I was setting up that field experiment in the static plots, I actually chose for half of those plots to be more dominated by blue grama and the other half of the plots to be more dominated by black grama. And we actually did find, let me see if I can find that slide for you. Yeah, here we go, this better slide. So here are just the static plots, and this is blue grama biomass, so not plant soil feedbacks, where it's in a static plot where it is rare versus where it's common. And you can see that the feedback strength, so the difference between its performance in black grama microbes or blue grama microbes, the difference between these two bars depends on relative frequency. And so there are these connections between plant soil feedback and the relative abundances of plants. It's hard in this case to say which caused what, because we can only correlate what we measure in the plant soil feedbacks to the existing differences in relative abundance and dynamics. And so in this way, it's only correlative. So we can't say, it is the negative plant soil feedbacks that caused the rarity of this species and also its stable dynamics.
A question we have is, if microbes aren't necessarily the source of stabilizing coexistence like you expected in the system, do you have any hypotheses for what it is, or ways you would approach finding out experimentally?
Mm-hmm. Yeah, that's a great question. That's something right now for the study in the sagebrush steppe ecosystem. I mean, certainly the thing that comes to mind is resource competition, the classic. So we have done some soil nutrient analyses to try to retroactively get at that. And it doesn't seem like there are very strong patterns in the differences of kind of relative availabilities of different types of ions and nutrient resources that you might imagine these plants partitioning. I mean, I think to more specifically get at that part, you would have to do some kind of competition experiment, which we actually did try to do with seedlings in the greenhouse, but there, we weren't able to get the densities high enough for there to be very strong competitive effects that to be observed. And then another thing that we've been thinking about for this particular ecosystem is the role of not just soil moisture availability, but soil depth and how that relates to moisture availability at depth. And that may be kind of a niche axis that we have previously neglected that could be important. But it's also something that's kind of difficult to measure well. Certainly when we've just done the very perfunctory stick a soil moisture probe in there and stick it everywhere and try to measure soil moisture differences, we haven't seen those patterns, but maybe there's something interacting with soil depth that we haven't really discovered yet.
A question we just got was, have you noted any above ground pathogens in this system? Could there be species specific foliar pathogens, for example?
Yeah, that's a really good question. And I would say for both systems, I mean, I've always just been keeping an eye out, broadly speaking. We've never directly tested for foliar pathogens, but it does seem like, especially in the New Mexico system, it's so hot and so dry. I've never seen any above ground signs of disease. In the sagebrush steppe ecosystem, it's also been the case, I haven't observed personally. There actually was a case where it wasn't a naturally occurring pathogen, but when we were growing out the seedlings in the growth chamber before planting them out in the beginning of the season, because of like the kind of cool, moist conditions of the growth chamber, some of one grass species started to develop this kind of mold on its leaves, but it was just like all of those plants did, and we just used the plants that didn't have mold on them. And it wasn't something that I saw again in the field on the same species. So I think that was a growth chamber condition issue. One thing that we did have trouble with in the field in the sagebrush steppe was grasshoppers, so herbivores. So my first season out there, when I was starting to plant out this 600 transplant experiment, I'd plant for 2 hours, come back, and half of my plants would be gone, right? Just completely devoured to the ground by grasshoppers. And so we ended up making a lot of grasshopper cages for all the individual little plants. And I suspect that although herbivore pressure seems strong in the system, seems unlikely for it to be a coexistence mechanism because there was no preference that I could tell to herbivore consumption, grasshopper consumption on these little transplants. They were just, the first green things out there, and they got hammered.
So one question I had, I know that since you started your professorship at UGA, you're thinking about the change in the ecosystems. And so I guess I wanted to know how you plan on adapting what you've done with grasslands into more forest ecosystems.
Yeah, I mentioned during the grad lunch that now that I've established my lab at the University of Georgia, in Georgia mostly there are forest ecosystems. There's not a whole lot of, prairies or grasslands. And one thing that I've been thinking about is not necessarily within the context of plant soil feedbacks, but one thing that I'm really interested in moving forward are the temporal and spatial dynamics of plant-microbe interactions. And so I started a new collaboration where we're really trying to figure out the temporal assembly dynamics of associated microbes as tree seedlings mature. And so there's been this unique opportunity in collaboration with the forestry folks here where we can do this in a very kind of large scale way that I could not have imagined as a field ecologist. So that's one of the hopefully new exciting lines of inquiry.
Are there any final questions in the chat? Please add them. I guess another question I had was about the ghost of competition present. I guess what plans, I suppose, do you have to, I think, remove that bias in observational studies and possibly applying that to more experimental studies?
Yeah, I think it's not necessarily a bias that you have to or can be removed. But I think going forward, the important thing to understand and note is that population models and competition models that you parameterize using observational data cannot be used to predict experimental outcomes, right? That's the kind of disconnect that's really happening here. And so I think moving forward, some of the questions that we have are, so if that can't be done, what are some of the ways that we can bridge this disconnect using different types of experiments potentially, or whether it's even like, should we even be using observational data, parameterized models to predict experimental outcomes? And maybe there are different ways to be able to test and predict plant dynamics, whether it is under climate change or other types of future scenarios that we can't mimic now.
That seems to be time. So again, Annie, thank you so much for a wonderful talk. Thank you, Beth, for hosting us on Zoom and YouTube. And thank you for everyone for your questions and chiming in.
Thanks, everyone. Bye.